Hi guys, welcome. If you have questions, please do ask away. Oh, I'm sorry you feel that way. What year are you in? Year 12 or year 13? Year 13. What topics have they been teaching you recently? It is tough. It is tough. I feel for you, honestly. You think it's easy when you pick biology and then you actually realize the amount of content that you have to do, and it's just unbelievable. Keep going, keep plodding. It will slowly start to come together, hopefully, to give you a bigger picture, but it is tough. It is so hard. Oh, you just finished cloning and biotechnology. That's an interesting one because there's lots of different parts to the cloning side. So you could be looking at the animal cloning, you could be looking at the plant cloning, you could be looking at the gene therapy side of it. It's really quite a, I like it. I like the topic area. I like teaching it, but I would say it's hard to get your head around because there's so much. I haven't actually done much on that one when it comes to videos, actually, to be honest. When I say that, I mean the lives. Whenever anyone jumps on the lives, they're always talking to me about the other topic areas, the module 5, etc. If I want to be a physics teacher at just GCSE, would I need to take it for A level? Right, okay, so no. If you are wanting to be a teacher, and so for example, I am a science trained teacher, I can teach all the 3 sciences up to GCSE, but I'm a biology specialist, so therefore when it comes to A level, I teach, or IB, I teach biology. I wouldn't even try to do physics or chemistry at A level because I don't have the time to get the knowledge or the interest, to be honest. And if the school's pushing for you to do that, that means they've not got enough teachers with the specialism. Do you have any videos on Hardy Weinberg? However, I do have videos on Hardy Weinberg. Yeah, I do. So it should be a video called Hardy Weinberg and speciation. But I can cover a bit of that tonight if you would like. You want to look at some Hardy Weinberg equations? Let's do that now. Now I am on my other channel for some reason. I can't get it to display on both. I don't know what's happened. It has decided all of a sudden it doesn't want to show you on TikTok and YouTube at the same time, so I need to have another play with it. Right, give me a second, let me think. You want Hardy Weinberg? I've just done my new study guide, which is on patterns of inheritance, and that actually covers the Hardy Weinberg content. So let me just get it up and bring it across. So let me come over here. I'm going to add a source. Bear with me 1 second whilst I get the size of this. No, that's not what I wanted to do. Give me a second. Let's go. There we go. Right. So, looking at the Hardy-Weinberg equation that I've just hidden from my screen. 1 second. Oh, I'm hitting all the wrong buttons tonight. and just see what I'm doing. Right, let's try it again. Second, did not like me doing that there. Let me just remove that and try it again. Okay, so we look at the Hardy-Weinberg equation then. So I want to go That's that one. That's that. Here we go. Okay, so whenever you're looking at Hardy-Weinberg, the most important thing to understand is the P and the Q and what it means. Because I'm doing GCSE this year, I'm picking A-level. I want to do physics teaching, but not do A-level physics. I'd say if you want to become a physics teacher, I'd be surprised. I mean, they're screaming out for physics teachers, so therefore I'd be surprised they don't want you to do physics at A-level, but it depends on the skill. If you go to a lower school, you'll be able to do it. Do you do predictions towards exam season? Yes, I would. I say predictions. I don't do predictions. I give you my best educated guess on what I have looked at. However, I will go through again. I am going through again all the papers and looking at what I know, what I feel like is coming up, and what I feel like you want to focus on. But it's the whole content. They could ask it in any which way, so it really just depends. And then if you want it for getting a 9-9 GCSE combined, what would it be? Make sure you've got your details and do lots of past papers. Absolutely agree on that one. Do you also teach GCSE biology? Yes, I do. I teach all the sciences. Although I don't tend to do a lot of GCSE at the moment, and the reason being is I am busy doing the A-level stuff, but I'm trying to do some videos out there for GCSE to try and help them, because I do feel like there's a lot of gaps that are missing from GCSE knowledge for students who come up to A-level. So that's where I would say we are at. Okay, let's have a look at this then. 1 second. Make that a bit bigger. That should fit down here. Okay, so let's do Hardy-Weinberg's equation then, and let's go through some of this knowledge. So as you can see from the revision guide, it's important to understand what P and Q actually talk about. So when we look at the genetic diagram, so I'm going to do a monohybrid cross, use that as the guide to begin with, and then we'll go from there. So if I have a capital A lowercase a, so I've got a heterozygous individual, capital A lowercase a, and if I've got that heterozygous individual, what are the possible alleles that the offspring could have? Well, the offspring could have any of the alleles. So I'm basically coming down and coming across, coming down and coming across each time. So I've got a capital A, I've got 2 capital A lowercase a, and 1 lowercase a. So therefore, because that's what I've got, I then want to use that and say, right, instead of saying A, I'm going to use P equals the capital A and Q equals the lowercase a. So if I do my allele frequency, I get P, Q, and I put that in my grid. And I put P and Q, and I put that in my grid. I'll end up with PP, which equals P squared. I've got a P and a Q there, a P and a Q there. And so therefore I have my 2 PQs and I have my QQ down here, which is my Q squared. So when we do, I can't put it on this screen, it is on my YouTube screen though. So if you want to jump over to Biology with Christine on YouTube, you'll be able to see what I'm actually writing with my study guide as well, that I have just finished. Perfect. I'll see you over there, and you can also write in the chat, and hopefully I'll be able to answer any questions you might have there too. Oh, you're very welcome. Sorry, I've only just seen your message, Thalia. Okay, so what is Hardy-Weinberg's equation? Hardy-Weinberg's equation is that p squared plus 2pq plus q squared equals 1. And what we also know is that p plus q equals 1. So if I have a population and I know that in that population I have got, let's say, 1,000 individuals. So if I've got 1,000 individuals and I know that, let's say, 4% of those 1,000 individuals have the, hi, oh, welcome Frankie. We're starting mitosis now in year 12. Nice, I love mitosis. So we got 4% of 1,000. So if I look at this then, if I know I've got 4% of 1,000 are going to be my q squared. What I need to do is I need to say, right, if I've got 4% of my 1,000, I need to get my calculator out. Calculator. There we go. Right. So 1,000 times my 4%. What have I got? So I should have known this. This is where my brain just doesn't like it. I've got 40 out of 1,000 that are going to have whatever this recessive trait is. So we know that Q is the recessive trait, and we know that it's the recessive allele. So if I've got 40 individuals, I would do 40 divided by 1,000, which equals my q squared. So it equals 0.04. So q would be the square root of 0.04. So if I can work out my allele frequency, so I want the square root, this is where I'm trying to use this calculator on here, and it's not going to give me what I want. There we go, 0.04. So therefore, Q equals 0.00, what did that just say? 0. 4. So we've got some really small numbers right now. That doesn't look right, does it? This is where a lot of students tend to make mistakes. So if we're looking at the number of individuals and we're trying to work out, and you start doing some calculations and you start to get really random numbers, you need to stop and think to yourself, well, is this making sense? Is this what I'm expecting it to give me? And if it's not, stop and restart. And a lot of students don't, they just keep going. What have I done wrong here? What's the problem with what I've put on the screen? What should I have done? So if we're looking at this and we're trying to work it out, are you doing live every day or just Monday? I normally do a live on a Wednesday. It depends how I'm feeling on the Mondays, but I'm not going to do Wednesday this week. I'm just going to do today, and then I'm going to stop for the Christmas break because it's getting to the point now where I need to have a bit of a break. My brain needs a bit of downtime, and obviously the girls are going to be at home as well, so I want to be able to have some time with them. So I'm going to have a break over the Christmas, and then I will be back for the New Year from the 5th of January because I am running my sessions on the 5th of January is my first year 13 session that I'm going to be doing. So that will be in January on the 5th. But then from then on, I'll do a Wednesday at 9 o'clock every week will be the lives that I do. So hopefully that will be helpful. But I'm still trying to work out, can anyone tell me what I did wrong? What have I done? Is it that your Q squared is actually your Q? I'm not sure. So what's wrong is because I'm using a calculator that I'm not used to, I've done the wrong part of what I should have done, okay? So I put Q squared equals 0.04 because that's what we worked out, 40 divided by 1,000. I've then gone Q equals the square root of 0.04. That's correct. But Q equals 0.0004, that's not the square root of 0.04, is it? And so this is where I would say it's really important you get to a point where you are using the calculator and you're practicing with the calculator that you're going to be using in the exam. So a lot of students actually would just write that rubbish piece of answer down and go with it. What is the square root of 0.04? So can anyone work that one out? What's the square root of 0.04? Because it's definitely not 0.00004, which is what the calculator gave me based on what I typed in. And this is the thing, like a lot of the time when we're trying to teach you a skill as a teacher, one, we may make the mistakes, or two, we're trying to get you not to just become this passive learner. There's no point in you being in these sessions and just sitting there expecting me to give you the information, because you'll never be successful in the exam. I'm not sitting the exam, you are. Absolutely, Frankie, thank you. So Q, when I do the square root of 0.04, is 0.2, isn't it? So if I know that my Q is 0.2, now I need to remember my equation. So P squared plus 2PQ plus Q squared equals 1. The other part of the equation is P plus Q equals 1. Thank you, Norva. So if I know that, then I need to now bring in, well, P plus Q equals 1. My Q was 0.2, so P plus 0.2 equals 1. So if I rearrange that equation, 1 minus 0.2, that will give me what P stands for, or the frequency of P, which is 0.8. So, excuse me, what I need to understand is there's an assumption. The assumption is that if I have got 1, which is P plus Q, if I know the frequency of Q, I can work out the frequency of P. Or if I know the frequency of P, I can work out the frequency of Q. And then I can bring that into this equation where, Johnny, no way! Oh, sorry guys, I have to be like, Johnny, if you're still here, oh my gosh, hi! One of my ex-students from a number of years ago. How are you? How is it going? How's uni? Oh my gosh, that makes me really happy to see you there. How are things? And I love the fact you called me a go. Johnny is actually one of the reasons why I'm doing what I do today. It was all down to him and Lara who suggested it. So if you're still here, I'm still going. Can you believe it? Right, we're going to substitute our P plus Q into the 1, and then we're going to do, if we want to work out P squared, we would therefore have 0.8 squared, which would tell us what the p squared is. Uni's so good. Ah, I still come up in your FYP. I love that. I love that. And I'm glad you're doing well at uni. I knew you'd be successful. I always believed in you, didn't I? Even when you didn't believe in yourself. 0.8, and we're going to square that. And that's going to give us 0.64 when we look at our homozygous dominant. This is my homozygous dominant. And then if I want to do my heterozygous, I do 2pq, so 2 times 0.8 times 0.2, which equals times 0.2, which equals 0.32. So if I then use that and I say, right, well, we had 1,000 individuals in our sample size, 4% of them were homozygous recessive. How many are homozygous dominant? How many are heterozygous? I would literally do 0.64 times 1,000 to give me the number of individuals which are homozygous dominant, and I would do 0.32 times 1,000 to tell me how many individuals are heterozygous. So let's just try that, times 1,000, so that would be 320, that would be 640. And then if we added them together, we should get our population size of 1,000. So when we do this, what we're looking at, and the reason we want to use Hardy-Weinberg's equation is because what we're going to look at with Hardy-Weinberg's equation is we're going to use it to work out if the, just zoom out slightly, there we go. If we're looking at the data and we've worked out how many are homozygous, how many are heterozygous, how many are homozygous recessive, what we can then do is we can look at, actually I want to go back, she says no, I'm going in the wrong direction. I want to look at the patterns of natural selection. So you need to know about 2 patterns of natural selection. You need to know about whether it is directional or whether it is stabilizing. So what we had there was 640 plus 320. All of those actually showed the dominant trait. Only 40 of them were showing the recessive trait. So therefore, what we can see is there is a selection which is a directional selection, and that directional selection is towards the homozygous dominant. So that homozygous dominant genotype has some advantageous condition that means that those individuals are actually able to survive against whatever the selective pressure is. So when we talk about selective pressure, whether that is the environment, whether that is the mating side of things, whatever it is that is a selective pressure, is it the conditions that are there that are selecting for? So normally we'd use the example of, let's say, the peppered moth. When it was the Industrial Revolution, it was more favorable for the melanistic peppered moth because they were camouflaged. As we have cleaned up our air, as we've got better reducing pollution, reducing particulates, we're moving in the opposite direction. What we would expect is we would expect to see a move towards, let's say, a recessive trait being more favorable, where there is an increase in the population size due to the fact that they are able to survive better, basically. Does that help at all? I know I've kind of gone in a bit of a random way there, but I'll give you an example of a question which links the two things together. So let's do this one. Okay, so if we look at this, sea otters. And this is one of the things that they could actually do looking at conservation. Oh, hi, Adam. Oh, thank you. Hi, Carol. Don't know if you're still in here. Sea otters, close to extinction. Close to extinction. Why were they close to extinction? Well, what was it that was happening? Well, there was a lot of hunting going on. So, if there's a lot of hunting going on, that's obviously going to decrease the number of otters that are able to reproduce, that are able to then pass on their alleles to their offspring. So we're going to see a decrease in the allele frequency within the population. So we have these sea otters that are close to extinction. We have a really small gene pool. And what's going to happen is, because of conservation, we're going to ban the hunting on sea otters. Now, the size of the population is going to increase. Obviously, nobody's allowed to hunt them anymore. They can then start to increase their numbers. We have sexual reproduction that's going on. We have random fertilization going on. We therefore get increase in numbers. And if we start to look at the frequency of the two alleles of a gene, we can then see what happens. So they'll look at one particular example. So in this case, we're looking at the dominant allele, T, which codes for an enzyme. And then we've got the other allele, lowercase t, which is recessive and does not produce a functional enzyme. It's that easy to apply. Honestly, it really is that easy to understand. And you only have to know 2 of the 3. So there is directional, stabilizing, and disruptive, but for OCRA, you don't need to know about disruptive. They've only named those 2 in the exams. Thank you for the likes, Adam. I do appreciate that. But that's the sort of thing where it's about taking the Hardy-Weinberg equation and putting it into context of why is it something that we're going to use and look at. So the sea otters, we've got these 2 functional enzymes, so they could go down the route of, well, what is it that causes it to produce a non-functional enzyme? So one way this question could go is it could look at protein structure, because it could look at the fact that it has a different tertiary structure. It could look at the fact that there was a mutation, depending on the way they want to assess you. But what we're actually asking is, if you've been told that the frequency of T is 0.2, so this time, instead of them saying 1,000 in the population and 40 of them had the recessive trait, now they're saying, right, well, T is 0.2. Well, T is basically Q, isn't it? So therefore, if we know that Q is equal to 0.2, then we go the opposite way. Q squared would therefore be 0.2 squared. So if we know, oh yeah, Safi, I'm on YouTube. It wouldn't let me do it up here. So yeah, that's the one thing you have to realize is sometimes they'll make you go a different way. So here we've got Q squared, 0.2 squared. So we're going the opposite way. So 0.04, and then we're timesing that by 100. Oh, crumbs, add an extra 0 there. And that therefore exactly would mean that there was 4% of the population that had this non-functional enzyme. So they wanted us to work out the homozygous recessive sea otters. So they basically wanted us to work out the Q squared. They could have changed that and they could have said, right, okay, work out the heterozygous. And again, once we know what Q is, P plus Q equals, try that again, 1, we just rearrange. So P equals 1 minus the 0.2, which equals the 0.8. So it's just another way in which we can use the equation, turn it around. But one of the things that's really fascinating is what they could then do is take it a step further and say, okay, if that's what you understand, can you then use that to draw a conclusion? So what type of natural selection appears to have occurred in this population of sea otters? Explain how this type of selection has led to a decrease in the frequency of the recessive alleles. So if we know that P plus Q equals 1, we know that P equals 0.8, we know that Q equals 0.2 even, then what we can do is we can work out our P squared, 0.8 squared. We can work out our 2PQ, which is 2 times 0.8 times 0.2. Norva, to answer your question, why did I times it by 100? Going back up here, I times it by 100 to work out what percentage of the population were going to carry those alleles, because it asked me in the question to do that. It said to work out the percentage of homozygous recessive otters in the population. So once I've got what Q squared equals, and I know that Q squared is my homozygous recessive otters, I therefore take that 0.04 and times that by 100. Now, they didn't tell me what the population was. So if the population were, let's say, 3,459, of the 3,459, 4% of them should be homozygous recessive. Because I've used Hardy-Weinberg's equation, and with Hardy-Weinberg's equation, we're saying that there is stability with the allele frequency. There's no movement. That's why Hardy-Weinberg's equation doesn't actually work in the natural environment. But when we're saying that there has been no factors that affect it, then we say that they're stable as the dominant allele is much greater in frequency. Or have I jumped to that? No, that's really important. You're very welcome, Norva. That's it. That's what I want you to go to. So we know that we've got our P squared 0.8. So that was our 0.64. We know that this one gave us our 0.32. So again, we can see that we have 64%, which are homozygous dominant. We've got 32%, which are heterozygous. And we have got the 4%, which are homozygous recessive. So therefore, we can see that there is a directional selection, and that directional selection is saying to us that it's actually disadvantageous. I don't even know, is that the word? It's not good to have 4% to have the homozygous recessive alleles. So you're not at an advantage. It's a disadvantage if you have those recessive alleles. So those individuals with those recessive alleles are less likely to survive. Because they're less likely to survive, they're less likely to reproduce. Doesn't mean they don't. And this is another thing you have to be careful with. It's that they are less likely to do it. So they're less likely to survive. They're therefore less likely to reproduce. Therefore, those alleles are less likely to be passed on. And that's why the allele frequency is decreasing. So I hope that makes sense. But Hardy-Weinberg, once you've got the equation, once you understand the way in which you can put it into practice, it's actually not that difficult. But they just love throwing ones at you all the time. So let's see if I can give you another example. Oh, excuse me. I've got the cold. I'm glad I had my flu vaccine because I'm not feeling as bad as I think I would be if I hadn't had the flu vaccine. But I just got my voice back and now I'm starting to feel like the effects of the cold. So I'm sure it is. Here we go. What's this one? Here's an old style one. Let's try another one. It helps a lot. That's all right. That's the whole point. This is the reason why I do these free lives, because I want to help students as much as I can. And whilst I am downloading my group tuition session, which actually has just finished now, the recording, whilst I do my editing and do that, I'm like, well, I have to stay up anyway. And so I want to help. So this is why I do it. Okay, we're going to do one more. Let's have a look. No, that one and this one. Okay, so what are we being told? We got Hardy-Weinberg's principle. We have the equations because they should give you the equation. A number of times I've seen it where they forget to give you the equation. Is quite interesting. So we're being told about our p squared plus 2pq plus q squared. So straight away, I would just write a little note to myself, homo dom, homo recess, hetero. So this is something that I would be doing when I've got a question, just to try and prime my brain thinking about what it is that they're going to ask me. So let's see what they've told us. We have albino rabbits that have got white fur because they're unable to produce pigment, melanin. The ability to produce melanin is controlled by a gene with a dominant allele. Right, so I know capital B, or capital B lowercase b is going to result in brown fur. So that is my genotype. That is my phenotype. And I know that the recessive allele, which is the lowercase b, I will need to have 2 alleles present. Otherwise, it will not be expressed. So this is where it comes together. Checking your understanding about monohybrid crosses, checking your understanding about inheritance, checking your understanding about how that links to Hardy-Weinberg's equation. So we're being told in the question that there are 60 rabbits. 60 rabbits in a pet shop. 45 of them are brown. So I'm going to take a new screen. Excuse me. I've got 45 brown rabbits. So therefore, I've got a total of 60. If I know that 45 are brown, 60 minus 45 gives me the number that are albino. So how many albino rabbits do I actually have? That would be the first thing to do, is to work out your recessive trait. We know that it was capital B, lowercase, which were brown. And we know that it was lowercase b that was albino. So we know that our albino is our q squared. So in total, there are 45 out of the 60. We should then hopefully know, thank you, Adam, that there are 15, good, which are q squared. So 15 out of the 60 equals, so we've got 15 out of the 60 that are going to equal our q squared. So if we then work that into which equals 0.25. So 0.25 is going to, thank you, Blaster Bro, going to be our part, which is Q squared. So we're happy. Is everyone happy with that point there? How did we get to that Q squared? We took 60 minus the 45, which gave us our 15 albino. Then we need to turn that into a allele frequency. So Q squared equals 0.25. So Q would equal the square root of 0.25. So that's the next thing we need to do, is we now need to square root it to work out the frequency of Q. So what is our square root of that? Let's see if I can do this the right way. It's like I want to use my phone. I'm like, where's my calculator? I really actually have no idea how to do it on here. So crumbs. Let's try that again. So Q equals, thank you. Oh, careful, Blaster Bro. Q should equal to 0.5. Oh, you mistyped. 0.5. So Q equals 0.5. So therefore, P plus Q equals 1. So P equals 1 minus 0.5. That's it. Well done. Well done, Norva, as well. So we've got that P equals 0.5 as well. So straight away, we've worked them out to be 0.5. And if we go back to the question, let's see what the question actually asks us. Using the Hardy-Weinberg equation, calculate the frequency of the dominant allele. Well, that's what we just did. We've calculated the frequency because we calculated it to be P equals 0.5. Now, what they would then potentially go on and do is, excuse me, ask another question like this. Okay, why is it not appropriate to use Hardy-Weinberg's equation? Why is this principle about estimating the frequency of alleles in these rabbits in a pet shop not appropriate? And this is where we need to look at, right, well, first of all, what did they have? They had 60 rabbits. So if they've got 60 rabbits, is that a large sample size or is that a small sample size? So always look at, in any question that they give you, always look at how many individuals they have told you in the sample. 60 is small. I would say ideally, well done, Frankie. Ideally, you want to have quite large sample size. So you really want between 500 to 1,000, but they can actually give you one that says 200, and you can say 200 is an okay size. It's definitely larger than 60, and 60 would definitely be small. So if I was sampling 60 people, or I was sampling 200 people, am I going to get a better representation of the population with 60 or 200 or 500 or 1,000? So it is about the larger the number, the better, until it gets to the point where actually it doesn't matter how large we go, the plateau in our data. But that's like a whole other thing. Let's not go down that route. So the sample size is a small one. So we need to be aware of that, in the fact that that's not going to show us the true population. Do you do live streams? Yes, I'm doing it now. Miss Aya, I'm on there now if you want to pop over. The other thing to think about, what are we told? We're also told in the question, and this is an interesting one. Oh, go away, that one. This is an interesting one in the fact that we're told in the question little hints. So always look for these hints in the question. We're in a pet shop. So because we're in a pet shop, we're in a pet shop, what have they told us? Well, this is not in the wild. Therefore, this is not random chance encounters, random mating. This is very selective. And because it's very selective, because it's not the wild, it's in a pet shop, we are therefore skewing the data. It's normally a Wednesday and a Monday if I can. I'm doing tonight, but I normally do a Wednesday, but I'm not going to do Wednesday this week because actually it's Alan's birthday. So we're going to, after I've tutored, have a evening to celebrate the birthday. So because it's not in the wild, because it's in the pet shop, it means that there is no random mating. And Hardy-Weinberg's principle only works if there is random mating that's going on. Now, what we also have to understand is that you go to a pet shop and you see that really cute little rabbit and you remove them and you take them home. They're no longer part of the gene pool. So therefore you've got this movement of the genes in the sense of we've got the gene flow being affected. Because we are removing some of the genes when we choose those rabbits, we take them away from the pet shop, we are decreasing the available genes for the continuation of the mating. The other thing to note as well is what colors have they got? We've gone brown and white, but how many different colors have you got? What shades are there? There's so many other things to take into consideration. So this is why Hardy-Weinberg's principle only works if we're looking at a dominant and a recessive allele, and not if we're looking at other factors that may have an effect. So you have to kind of bring in lots of different things. But remember, if you ever ask a question on this, they're only looking for 2 reasons. And because they're only looking for 2 reasons, they want you to only give 2 reasons. They never want you to give everything because you're not going to remember all that stuff. You're going to, one, write it in your way. You'll notice it when I'm talking right now. I'm getting tired. It's been a long day. But the way I say things, sometimes I'm absolutely on it and I can articulate myself really well, and then other times maybe I'm not as articulate as I should be. However, should I be penalized because I've written it in a very high convoluted way, or should I be penalized because I've written it in a very layman's term way, but I've still given you the information that you require? And I think it's important to note that when you are writing your answers, don't get hung up on the examiners expecting you to write it in one specific way. Don't get me wrong, there are keywords you must say and you should use and you should have learnt. However, if you don't know those keywords but you describe what it is that should be happening, then you will get some marking points if it is correct. If it's wrong, it's a little rubbish, obviously you get nothing. But if it's correct but you've just described it in a slightly different way to this word, that word, this word, they're not going to penalize you on that front, because there's what, 20,000 of you sat it last year. There's probably going to be about 20,000 of you sit it this year. You're not going to answer exactly the same way, and so therefore it is important that you don't get hung up on, I'm not smart enough to be able to get this. As I talk right now and as I listen to myself, I'm like, oh my gosh, I sound awful tonight. One, I'm tired, I've got a sore throat, I'm not feeling the best. That's fine. But at the same time, I have a master's from Cambridge. I have a postgrad from Cambridge. I actually have 2 postgrads from Cambridge, and I also have a undergrad degree. And yeah, I'm fumbling tonight. I'm making mistakes. That's okay. It's human. It's natural to make some mistakes. There will be some leeway, and you'll notice when the examiner is and giving you the benefit of the doubt, they'll have what's called the BOD, or they'll go NBOD. So BOD means benefit of the doubt. Do you know what? They haven't quite given me exactly what's on the mark scheme, but I'm going to give them the benefit of the doubt because they're going along in the right direction. Or absolutely not. They've not given me enough. So although I've read it and although I see where they're going, I'm not going to give them the benefit of the doubt because I don't believe they really knew where they were meant to go with that question. And so that's what I would say. Nicomantu, it's not a class as such. This is where I offer free lives, where I jump on here and I help any student who is struggling with their A-level biology. I do live class. I do actual classes that are not like this. This is at the end of me having taught and done a lot of stuff today. But my classes that are actually planned and conducted, that are paid sessions, they are not me just answering questions on whoever's on TikTok or YouTube at the time. So yes, you've joined a kind of class, but it's not a official class. It's more just me chatting and helping anyone who's stuck on A-level biology. Because I get it, not everyone can afford tuition. That shouldn't stop you, but also at the same time, I also want to make sure that the level of content that's out there is high enough to get you up to that A-star grade. Because I've got the training, I had the years of teaching, and I want to give back. And so that's what I'm doing. But I am going to stop now because I'm getting tired. I can tell I'm tired. I'm definitely ready for the Christmas break. But if you are interested in joining my group tuitions, I do them on a Monday evening, starting the new one in January. On the 5th of January, there's 5 spaces left on that one, I believe. So yeah, if you're interested in that, then do send me a message on here or on here. Or check out my website, ukbiotutor.com. Is so unaffordable for some. 50 an hour. I have to say, Frankie, that is actually lower than my hourly rate for one-to-one, but my group tuition sessions are £25 an hour, and they are small group sessions. And so I have my one-to-ones with those who really need targeted, and obviously they're the ones who pay for me to be able to do this. But also I do offer my group tuition sessions at a reduced cost. But the thing you have to remember is, for those of you who are looking for tutors, I would advise you be careful for the cheap version of a tutor that they have the credentials needed to get you where you need to be. I had someone who joined me last year. They were, let me get this right, 7 weeks away from their exam, and they had been with a tutor for a while, and they had actually been given wrong information, stumbled across my profile, came to see me, and we worked together for 7 weeks and they got their A that they needed. No, I know, Frankie, I'm not taking it, please. I'm not taking it as shade at all. I completely agree. That I'm actually not as expensive as some people are out there. However, the amount of experience that I have, you can spend the money on a university student teaching you. You can spend the money on a qualified teacher teaching you. You can spend the money on an expert. It really just depends on where your level lies. And I do feel that there's an avenue for each of you depending on what it is, but this is why I do my free lives, why I offer these sessions, because I know not everyone can afford it. And actually I'm working at a deficit right now. I took 6 months out getting no pay, building the YouTube channel up for students to have it, and I actually don't meet minimum wage when you look at how many hours I actually am able to teach. And so you've got to look at all of the different areas. But I would say be careful who you select. I'm not saying they have to be expensive. I'm not saying they have to be cheap. What I'm saying is make sure whoever you do choose has the right credentials to help you be successful and doesn't teach you the wrong misconceptions that you then take into the exam. Because if you take them into the exam, you are then at a disadvantage because you believe you've been taught the right information and actually you haven't. It depends, Frankie. I'm not going to say yes or no to you with regards to, for some students, you can do it on your own. So I had someone last year who did, how many sessions did they do with me? 5 sessions over the whole time period, one-to-ones. Every time they did some questions and they struggled, they'd send me a message. They'd have an impromptu session with me. We'd go through their specific questions of what they were struggling with. And they just attended my lives every week for free. And they got the A star at the end. So it really just depends on where you're at, what work you're doing. So if you're only predicted a C, just to be clear, a C is not an only predicted. A C is a really good grade, but is it what you want? If you want to get higher than a C, what is it that's holding you back? What is it that you need to do to move forward? A lot of students want the grades or think they can get the grades by passive learning, and that's not the way it works. Now, I can tell straight away that you want to learn, you've asked the questions, you're willing to try. So if you keep going with that and you keep doing practice questions, you keep asking questions on topics you're not sure about, you will then start to improve. But you have to be constantly doing the recap, constantly going back and double checking stuff, because if you're not, you'll forget it. And that's the worrying part. There's so much in A-level biology. It's like, where does the time come from? And it's about being smart with your revision. Being smart that when you're revising. So for example, as I was saying to those in my group tuition session tonight, there are 20 weeks left until the May half term of my group sessions to do with them. So it's 20 weeks, and in those 20 weeks, I've already planned out that I'm going to cover all of module 5 and module 6 specifically, because there's a few module 5 I need to break into 2 hours. There's a few of module 6 I need to break into 2 hours. And so for 20 weeks, I will cover all of module 5 and module 6, which then is like, oh yeah, that's great. However, what I will also do is I will also be dripping in module 2, 3, and 4 as I'm doing module 5 and 6. And that's what you need. You need that bigger picture, not just I'm doing module 2 and that is all I'm revising right now, because biology is the bigger picture. So if you're predicted a C, what are you doing right now that's holding you back from getting that grade? Or if you are getting that grade, what more could you do to get the next grade up? I would say, and I say this to every student I've got, aim for the A star regardless of where you want to be. Aim as high as possible, because if you aim high, you're going to be working towards that higher grade that therefore is going to help you to push past that lower bar that you may have set yourself. So a prediction is a prediction. It's not where you're going to get to, but it's about being realistic in, one, have you got the time, the motivation, and the energy to work out what you need to get that high grade? Or are you just spread too thin? And the other thing is, make sure you have a break. Make sure you take the time to have a break. Because if you don't have a break, like right now, I know I'm getting tired. My brain's not functioning as well. And therefore, I need a break. I know that. So I'm going to accept that. I'm going to take that break. I'm going to have a couple of weeks off. And I'll be back at the end of the Christmas. Well, depending on, I might have had enough of the girls and stuff. I might pop out here and have a one night random session. I don't know yet. But it's one of those things. Have the break. Give yourself the downtime, because your brain will thank you for it. You'll thank yourself for it. But then be ready to run when you start back in January. Because honestly, when you look at that 20 weeks, that's it. And then that's 4 weeks left. 1 week and then your exam is on the Thursday the 4th. Then you've got the exam on the Friday. Then you've got the exam, I want to say, on the Wednesday. I might be wrong on that. It might be the Thursday the following week, but that's how quick it goes. Right, I am gonna go. I look forward to seeing you guys in the new year. If I am on here, then please obviously jump on, say hi. But if not, then have a great festive period, and do put comments on my videos. If you watch any of my videos, do put comments on it. Oh, you're very welcome, Adam. Because I do tend to reply. If I miss them, I don't mean to, and I will come back and do it. But yeah, have a good one and enjoy your evening. You're very welcome.